While we're waiting on Dr. Lykina, I wanted to share a little bit of information about some other available recorded events that you can ask your admissions representative about. I know it's part of the requirements for when you're applying to OUM to make sure that you have a good understanding of our programs and have had a chance to meet some of our graduates and deans and faculty and other OUM students. And so these recordings are available to you. So for example, we did a Meet the Dean session with Professor Ross Diplock, who's the Dean for Australia and New Zealand, and who's also the Director for Clinical Education at OUM. And he gave us some really interesting details about the changes to the clinical program and what's upcoming for improvements in 2022 that you as prospective students, applicants and matriculants will be able to take advantage of. So I do highly recommend that lecture with Professor Diplock. We also have a Meet the Faculty that we ran a couple of months ago with Dr. Ahmed Musin, who gave a lecture from the renal system, which is in our system-based modules in the second year. So Dr. Lykina is going to give us samples of some information from our E-Foundations that happened in the first year of the preclinical years. And you might also want to have a review of Dr. Musin's lecture, which is from the second year of preclinical studies. So Dr. Lykina, I see that you're back. Do you have access?
Oh, thank you. Yeah.
Would you like to take over?
I'm sorry about this technical issue.
Not a problem at all.
Yeah, well, you know, some things happen when they're not expected. And
roll with the punches.
Yeah. And can I start?
Yes, please. I'll stop my share.
Okay, thank you. Thank you very much. I appreciate that a lot. So, hello everyone, then again, welcome to OUM, and thank you, Dr. McGuire, for so nice presentation and introduction. And today we're going to talk about the immune system. As you understood, I think already, I am a fan of immunology. I love it extremely, and I will try to do my best for you not to be afraid of that, because usually there is an opinion that actually immunology is something difficult, something very much complicated. Maybe it is, I can't deny, but actually it is really fascinating and explains so many things in our life. So there will be, I can't see, Dr. McGuire, I can't see slide. Still cannot. Is there a presentation you're sharing? So let's go on, let's move on. And today we're talking about the immune system recognition of self and foreign, because everybody understands, everybody knows that immune system first and main function is to recognize self and foreign, to kill the antigen, the aggressive pathogen, and to be tolerant, so non-reactive, not aggressive against cell tissues. And how it happens? How do you know that you is you? How will you understand that? How your immune system knows that? How to differ between the foreign and self? Ah, there it goes. Yes. And thank you. And now you see, it's not by chance I put these two cups on the first slide. At the end of the lecture, you will understand why is that. Because there are two actually receptors, presenters of self and foreign. And as far as they look similar, they function in the same way, presenting, but at the same time they're kind of different, because you see, one cup is full of tea, another cup is full of coffee. So there we are. Let's find out where is the similarity, where is the difference, how it functions and serves our purposes. What's the purpose? The one always for our body is to keep constancy, to keep homeostasis and function properly, protect from the surrounding. If you would like to show us the next slide, please. Yes, thank you. So there we are. Why HLA, and you see in brackets MHC. What these abbreviations do mean? It's always like a kind of a mystery when you go to abbreviations, to be honest, no matter what kind of abbreviation is that. And you always think, okay, what does this mean? And you see here, I put the explanation of that. So these two terms, HLA and MHC, they are synonyms, and they can be used interchangeably in our conversation today. Interestingly, I think that if you already connected to medicine somehow, if maybe you're nurses or maybe you're paramedics, you studied medicine a little bit, or maybe if you were interested already in medicine, you in many books were reading and seeing this HLA or MHC. And HLA is human leukocyte antigen, and MHC is major histocompatibility complex. These two terms mean the same, mean the interaction between the cells and receptors of their immune system and recognition self and foreign. Please, we should go to the next slide, Dr. McGuire. Thank you. So now it's the example from the very beginning of a teamwork, dear future colleagues, dear students. Here we are. We always work as a big team all together. And this is the first example. How it works at OUM. So major histocompatibility complex, and next I will use MHC or maybe HLA. It consists of genes and antigens, in other words, receptors, which are encoded by these genes. Everything as it is in our body. We keep information, how it should be constructed, how it should look like, and how it should function in our genes. And these genes control our physiology, the structure of our proteins, receptors, and everything in our body. Here is all the same, nothing new. And these genes, which control the receptors, antigens on the surface of the cells, they occupy chromosome 6. And they include 3 classes. The class 1 includes loci ABC, class 2 includes loci D, which also divided into DP, DQ, DR, and class 3 includes genes which determine the structure of complement components, such as C2 and C4, propedin, and factor B. And it's always important to remember some tiny details. And sometimes I can tell you need only learn by heart. You need simply to memorize. But there are some tricks how to memorize that. And the main and the best trick how to memorize is to understand the background of that. So how to memorize what class includes what loci. At first, when we were just starting understanding that there are such genes, and when the scientists understood that there are 3 classes and there are loci in those classes, we need always to classify, we need to give the name. And usually what we use alphabet or numbers, right? So we said, okay, there is a class, there are loci, there will be A, B, C. Then when research goes on, time passes and we see, oh, there will be also class 2. How to name it? What's the next letter in alphabet? D. But then suddenly we see, or maybe not suddenly after some research, we see that there are also other loci and decided to name them as well. And understanding that there are other loci already in class, decided, okay, let it be D-related. So it will be DR. And guess what? P and Q stand beside letter I in alphabet. There we are. That is why loci I in class Y includes loci ABC, class 2 includes loci D, DP, DQ, DR. Now, when you understand that, you will never forget. It's very, very understandable and very easy now. Please, next slide, Dr. McGuire. Yeah, so this is the illustration of what we've said before, because of course it is very, very easy to understand when you visualize that. Have you read the book of Rhonda Brown, The Secret? Or maybe you've seen the film. And one of the tricks is the visualization. So there we are. We make the picture. And look at this. You see here, class 1, which includes loci ABC, and it controls the structure on the surface of the cells in our body, the antigen receptor, which will present the self-substances, self-proteins. You see it on the right lower corner, and you see that these loci ABC control the structure of one type of the receptor. Another class, class 2, if you go to the left, you see DP-DQ-DR, this is what we've just talked about, and they control the structure of other type of the receptors, which will present foreign substances. This what attacks us every second, every minute, our body. And this what every minute task for immune system to divide between self and foreign. And you see in the middle, the third class, and these genes, they control structure of C2, C4, factor B, and also some cytokines. If you see the TNF, what does this mean? Because these all abbreviations, they are very scary from the first view. It is tumor necrosis factor. It is a cytokine which participates in different immune reactions. And I'm just looking forward to January to start our course of lectures, and we will talk about cytokines definitely as well. So we're moving to the next slide, please, and we will talk about the genes. So there we are, and you see now we only simply named the basic loci of these three classes, but of course, we're still, and scientists are still working on that. And now you see that there are more loci now, A, B, C, and H, and G, F. In class 2, there are more. You see DRB, DRA. So we're even running out of letters to name all these loci in different classes. So it is very much complicated. Why I've decided to give these slides separately, because, you know, it might be seen that I'm repeating that just information. Well, you know, it is really very important, because at the end of our lecture, we will talk about polymorphism and polygeny, and we will recall that there are so many loci in each gene, in each class of this MHC genes structure. It is really very important, and later today we will say why it is so needed, why it is really critical for our survival, if you like. And please, the next slide. And now we are going to talk about the antigens, receptors, which are controlled by these genes. And this, I modified this slide. It is the combination of different, you see, pictures, and I put the arrows so we see exactly what gene controls what loci of the gene controls what part of the antigen. And let's start from class 1. It is on the right side. You see its structure. We will go there in details, and it is composed of 2 chains. And you see that the genes loci A controls one part of this alpha chain, alpha 1. B and C controls alpha 2 part of alpha chain. And if you go to the left, and you will see that the same happens with genes and antigens, their relationships. And you see that DPB and DQB control the B1 part of beta chain, and the other loci, they control alpha chain, alpha 1. Why it is so important? Because actually the when you will study even the genetics as well as immunology, you will see that there are some genetical pathologies, and there are immunodeficiencies or in immunocompromised individuals who inherited some pathological genes, or maybe there was a mutation. Maybe it's not inherited, maybe it is congenital. It is possible as well. Inherited, it is from previous generations, pathological gene. Congenital, when the mutation happened exactly during the development of this fetus, this individual. Anyway, they will be born with some pathology, which is in genes. It is very difficult to make the corrections and to treat, but we are to understand why it is so much important. That is why we're giving this slide and we talk about that. And let's focus now. So we slightly move from genes to antigens, to receptors. So now we see the whole picture, how it is organized. And let's move to the class 1 antigen into the class 1 receptor on the surface of this cell. Please, next slide, Dr. McGuire. Yeah. Also, in other books, you can see that it is class 1 MHC, not antigen, but molecule. In some books, you will see class 1 MHC receptor. They are all terms are eligible, and you can use whatever you prefer. With antigen, molecule, receptor, we name the structure on the surface of the cell, which will help immune system to recognize the self of foreign proteins. And class 1, to tell you immediately now, that it recognizes self structures. The class 1 serves for the recognition of our own cells and tissues. And we already know that this receptor, this molecule, this antigen is encoded by MHC A, B, C genes in class 1. It expressed on all cell types except erythrocytes, adipocytes, and trophoblast. Why is that? Erythrocytes, they have a short life, so they're not very much needed to control how they function, because they actually even do not have nucleus, and they will be controlled by spleen. So, okay. Adipocytes, well, they're fat tissue. It is fat cells. Fat tissue, not very much recognized because no need. It also has a specific structure. You will study that when you go to the histological part of your studies. And trophoblast actually, it is a very interesting cells and tissue, which you will talk about when you talk about pregnancy. And pregnancy is a specific state in physiological as well. So there will be no need to control that much. Why? Because to keep the fetus for 40 weeks to grow up and to form its own tissues and cells, which is 50% foreign genetically. So it's kind of complicated situation for their immune system. That is why on trophoblast, it is not very much needed, this MHC. for certain reasons. But other cells, all other cell types, they all express MHC class 1 on their surface. And it is logic if you think that we need to recognize if our cells and tissues, our own cells and tissues, have normal structure. They're not mutated, they're not somehow transformed into maybe some malignant cells. All of them should be controlled, don't you think? So that is why all cell types, actually, immunology is very logic, and if you think from the position of protecting yourself, everything will become very easy. As we said several times already, it's major determinant of self-recognition, principal antigen recognition by the host during tissue graft rejection. We will talk about that a little bit later. And presentation of an antigen self protein to CD8 plus lymphocytes. They are cytotoxic T lymphocytes. And when you will join the lectures in January, when we start in January, teaching me and Dr. Boone immunology, we will talk about these cells separately. So looking forward for that. Next slide, please, Dr. McGuire. And we're going to talk about the structure. So we know the function, what it serves. So what's the structure of it? And then again, you see the same picture. We already have seen that in previous slides. And it has extracellular portion, transmembrane portion, and intracellular portion. And you see here on this picture that there are two chains, alpha chain and beta chain. Alpha chain is a very big, it has 3 compartments, alpha 1, alpha 2, and alpha 3. And to stabilize this huge structure of alpha chain, we use beta chain, and it is 2-beta microglobulin. Why? Because it's very small, it's micro, especially if you compare with alpha chain. And they all connected one to another, and in between the alpha chain with disulfide bonds. And you see here on the top of our picture, you see peptide binding cleft, as if it is kind of a cup, right? Shape of a cup. Remember the first slide why I put the cups? This is the association. So this cup is for what? For self-structure, for self-protein, or maybe some other, usually it is protein, some other chemical structure. So we put into this dish, into this cup, self-structure and give it for the recognition by the immune system. We say, okay, immune system, look how it works. What's the structure? What's the function? And the immune system will come and recognize and decide, is it self or foreign? And you see here that these parts of these chains, they are hydrophilic, hydrophobic, and there is a very, very short tail inside the cell and a huge part outside the cell, because the main function of recognition of our own antigen, our own structure, our own protein will be outside the cell, because we need to show it to the immune cells, these CD8 plus T lymphocytes. And please, the next slide. So we're going to talk about class 2. And as we already know, it is encoded by class 2 genes, this antigen, and we remember that it is DP, DQ, DR, and it is expressed on immunocompetent cells like macrophages, dendritic cells, B lymphocytes, and a little bit on T lymphocytes. And it presents foreign antigen to T helper cells, which carry the CD4 receptor. And this will be the mechanism for recognition of foreign substance. So let's remember class 1 for self, class 2 for foreign. And you see here that not all cells are able to present foreign antigen, because to present foreign antigen, you need at first to process it. You need to be able to interact with this foreign antigen, with bacteria, virus, anything else. You need to have some specific tools, if you like, to at first to catch this antigen, foreign one, the infect, first of all, of course. You need to be able to divide into pieces, and then to take one piece and to put it into this MHC class 2 receptor as the dish and to present it to T lymphocytes. So not all cells in our body are able to do that, not all. And it's only immunocompetent cells, which actually, with the first 3, which you see there in the brackets, macrophages, dendritic cells, and B lymphocytes, they form the group of antigen-presenting cells. So they can interact with foreign antigen, process it, and using MHC class 2 receptor, present it for the recognition by their immune system. Please, next slide. And of course, now again, the same. What we've talked about, what's the functions, now let's look at its structure. And you see quite similar for MHC class 1 receptor, right? Remember those 2 cups? They are very similar, but still different. So you see that MHC class 2 actually is composed again of 2 chains, alpha and beta. But these 2 chains both are big. They both have extracellular, transmembrane, and intracellular portion. These 2 chains both have 1 and 2 parts, and you see it on the picture. And then again, look at the top, peptide binding cleft. So again, you see this cup, this part, which will interact with the foreign antigen, keep it connected and presented to the immunocompetent cells, to T lymphocytes. The MHC class 2 will be presentation of foreign antigen. And then again, you see that they are also hydrophobic, some hydrophilic parts, and then again, the biggest part is extracellular one, because recognition and presentation will happen outside the cell on its surface. And only the terminal and intracellular portion is quite small. And let's talk now how it works together. So we now, and please, Dr. McGuire, the next slide. Yeah, let's compare how it works all together. And now this is another slide, another structure. Very, very quickly, we will go through all of that. So this is it, how it is presented on the surface of the cells. And we now know that MHC class 1 antigen is presented on the surface of all cells except a few. MHC class 2 only on the surface of immunocompetent cells who can present antigen. And this is the structure. And very quickly we repeat it, we compare that. Again, remember those cups. And let's go to the next one. Yes, a little bit busy slide, don't you think? But this all shows how it works. And look, let's start then again with class 1. And now on this slide, it is on the left side. So what's happened? Some cell may be infected, and we will talk about that, or maybe it is normally functioning our own cell. It presents self-antigen. You see this blue circle, it is self-antigen, or maybe it is some viral protein, or maybe it is mutated cell. So this protein is presented by class 1 MHC. Who will recognize? You see on the bottom, on the left side, left lower corner, CD8 T cell, CTL. What does this mean, CTL? It is cytotoxic T lymphocyte. And this cytotoxic T lymphocyte will recognize with a specific receptor, which is called T cell receptor, that it is TCR, combined with CD3. We will talk about that in January in details. And this is the whole complex of receptors. You see, not that simple, presentation and recognition. There should be the whole complex of receptors which keep the cells together, close one to another. One presents, another one recognizes. And look, there is a phenomenon of double recognition, which is very interesting. At first stage, at first step of this recognition, CD8 receptor, you see it on extreme left, these two sticks, they recognize MHC class 1 receptor. First step of recognition. And then we'll go on the second step when T cell receptor, TCR, recognizes this antigen, which is presented by MHC. Always 2 steps. If we go now, how it works for MHC class 2, you see then again that they needed the whole complex of receptors. And then again, double recognition, 2 steps. CD4 interacts with MHC class 2, the first step of recognition, and T cell receptor, then again, recognizes antigen, this blue thing, which is presented by the MHC class 2. And now it is foreign antigen. And you see here what cell? Antigen presenting cell will express this MHC class 2 with foreign antigen on its surface. So phenomenon of double recognition. At first, MHC is recognized by CD8 or CD4, and then only T cell receptor will recognize antigen. If it is MHC1, it is self. If it is MHC2, it is foreign. Please go to the next slide. And how to memorize, because actually, to tell you the truth, you need this to memorize, learn it by heart, this table. But it seems to be very easy. But to tell you the truth, because I have decades already experience of teaching, and I can tell that at first when we study it, when we have lecture about it, we understand that it is very easy. But later, when we begin to combine all the things, all the processes together, students usually are very much confused and make mistakes. So how to memorize? I give you the clue. You can use whatever you want, whatever your logic, how it works for you. But for me, it works the following way. Imagine there are 2 children in the family. First one is elder one. He or she is already 8 years old. The second one is younger. He or she is only 4. And then you will never forget. As I said, you can use my trick, you can use yours, or you can simply just memorize, learn it. Okay? So it's up to you. But this, what we definitely never ever should make mistake. We never should be, hesitating that MHC class 1 interacts with CD8, 2, 4. And then again, it's easy to remember, MHC1 presents self, it is internal pathway, and MHC class 2 presents foreign antigen, it is external pathway. And we're going to next slide and going to talk about these pathways. What are they? What does this mean, pathway, internal or external? Here is the one slide to compare, and we will talk separately in details about the internal and external one. And look, at first, the upper part on the green background, it is internal pathway. How it works? It is cytosolic protein, so it is in cytosol, somewhere inside the cell. Where it comes from, we will talk about it next slide. So what's happening just generally? Yes, they have the general idea. What's happening? The cytosolic protein will be divided into pieces, will interact with MHC class 1 in endoplasmic reticulum, and will be transported in the vacuole to the surface of the cell. And remember that there are all cells in our body. This is how it happens. In extracellular pathway, it will be, you see, digestion of protein. So the foreign antigen enters into our body. The antigen-presenting cells, not all cells, only those immune cells who can phagocytose, who can swallow this antigen, then will digest it, divide into pieces, then again in endoplasmic reticulum, these pieces will interact with MHC class 2 because it's foreign antigen, and will be transported on the surface of antigen-presenting cell and will be recognized by T lymphocytes. And we're going to the next slide, please. So in details, now maybe too busy, but we're not going to talk about the specific details, right? But look what's happening. Our genes, right, keep information about our structure and function. And due to the information of our genes, the cell will produce different compartments. First of all, proteins, of course. And normally, these proteins will be in cytosol. Some of them will interact with MHC class 1 and presented on the surface of the cell. And you see on the right side, blue cell, CD8, cytotoxic T lymphocyte, comes and recognizes, first step, MHC class 1 CD8, then self-antigen by TCR, by T-cell receptor. If cell is okay, not infected, functions well, T lymphocytes will not become cytotoxic. They will be not activated. They will recognize it as cell functioning well and will stay tolerant. This how immune system every minute, even now we're sitting talking about it, controls everywhere how we function. But let's imagine that cell is infected and infected by virus. And you know very well in terms of COVID now that these all viruses, they go where? They put their genetic information to the DNA of the cell. And now you see on the left side, you see the genes and you see the red color. So this red color is viral. And of course, according to the viral information, cell will produce viral proteins, viral particles. And they are in cytosol as well. And they will interact with MHC and present it on the surface of the cell. And T lymphocytes come, first step of recognition, MHC class 1 and CD8, everything is okay. But then when TCR recognizes an alarm, foreign, we do not recognize this protein, it is foreign, it should not be here. It is pathology. T lymphocytes are activated, begin to kill the cell. Imagine another situation. Imagine if cell mutated, transformed into the malignant cell. Also, of course, will be abnormal structure of proteins because it's not our own already, not functioning in a normal way, let's say. And this abnormal protein will be presented in complex with MHC class 1 on the surface of the cell. T lymphocytes come, recognize first step, okay. Second step, T cell receptor recognizes alarm, foreign, not ours, not proper structure. No, this cell doesn't function well. Let's kill it. Let's get rid of it. So this is what happens. This how immune system recognize our own cells which function normally, those who are viral infected, those who are mutated and transformed into malignancy. Another situation still in this slide, imagine transplantation. And remember we said genes control the structure of MHC, and we are absolutely individual in our MHC structure because we talked about the whole idea how it should look like, right? But all this sequence of all these alpha chains and beta chains, they're very individual in us. We are unique. Everyone is unique in their structure of this MHC. And imagine that the cell organ, let's say kidney, from one person with some certain MHC structure is given to recipient whose T lymphocytes are expecting to see certain structure of MHC. And what will happen if we are not accurate in finding the proper match of donor and recipient? Kidney from another person produce some MHC structure. T lymphocytes expect the MHC structure to be certain, right? And when they see some other structures, maybe slightly different, they recognize it as foreign. So the first step of recognition even will be a problem. That is why there is a rejection. That is why we need to give the immunosuppressants to depress the immune system. We need to find the proper of donor and recipient. HLA typing is used. That is why we look at first for the better match in between relatives, because we inherit from our parents and grandparents these genes, so we hope that it will be more match. However, it might be matched with some stranger. It is possible. So this how MHC class 1 works. Okay, now let's go to MHC class 2. It is a little bit more simple. Yes, thank you. So how it happens here? Just you see on the left side the protein, some antigen, bacteria, virus, maybe at the beginning, maybe protozoa, whatever, maybe some chemical structure, who knows. It will be phagocytosed, divided into pieces, processed. These antigens, small parts, which are called epitopes, they will interact with HLA class 2 or MHC class 2 receptor presented on the surface. CD4 comes, 2 steps of recognition. MHC class 2 recognized by CD4, foreign epitope recognized by TCR. And this is the beginning, the push of the immune process. This what we usually see how immune system works when it is some infection. There will be production of cytokines, activation of other cells, but this is all we will talk about in January. And next slide, please. Let's talk about the MHC restriction. As far as I remember, Okay, another slide. Then again, very quickly to compare, right, all of these functions. You see class MHC1 pathway, class MHC2 pathway. So how it works. The immune system recognizes self and foreign, stays tolerant for self if it is okay, and being aggressive against some abnormalities or foreign antigen. And now I think next slide. Yes, okay. Finally, MHC restriction. What does this mean, restriction? You see here on this picture that MHC interacts with the antigen. It holds it, really holds it to present to T cells, and T receptor needs to be compatible to MHC to recognize this antigen. And T cell receptor, CD8 or CD4 receptors, and CD3 receptor, they constitute the whole complex from one side. MHC, other additional co-stimulating molecules from another side. But the key, TCR and MHC. They need to be compatible. They need to interact completely in between together to recognize the antigen, no matter self or foreign, no matter. And if we go to the next slide, imagine what if there is no compatibility? What if MHC can't be recognized by this complex of receptors? And you see here is a funny picture, you know, because we were talking about the serious things. Let us relax a little bit. And you see, this is the dendritic cell, one from 3 types, which presents antigen. It presents with MHC class 2. And T cell says, sorry, but I don't recognize that MHC peptide. And there will be no recognition. What does this mean? No recognition. This means that viral infected cells may survive and produce virus. Tumor cells will survive and make lots of problem for the body. Foreign antigen will be not recognized. And if it is not recognized, if immune system is not providing the immune response, that's the paradise for antigen, right? Do what you want in the body. So there will be disease non-stop and even death if there is no immune response. So the army is sleeping and the enemy is invading. There we are. So MHC restriction is really very important thing. We need to understand that we depend on structure of MHC, and MHC structure depends on genes. Thus, let's go to the next slide and talk about...
Lykina, can I interrupt with a question?
Sure, yes.
Because I'm really appreciating this clarification of difference between class 1 and class 2. Like it really helps bring that forward, a really complicated process and a really complicated set of molecules, as you were showing with the genetics at the beginning. So I was wondering if you could tell students a little bit about the other kinds of supports and materials that they would receive when they're in their eFoundation. So like, for example, you know, this content is complex, as is all of the content in the basic medical sciences through the eFoundation. So like, for example, would they have access to slides and recordings? What would that look like for students in a week of eFoundation?
Yeah, thank you for the question, because that's really interesting to know. Okay, this was the lecture. What's next? Do I have access to that? Yes, you do. First of all, you see that the session is recorded, so you always can go and listen to the recording. If, for instance, you had no chance to be there for the live lecture, you can listen to the recording. Also, these slides will be downloaded, so you will always have access. They will download it prior to the lecture. So there is a syllabus. The whole, chapters are explained. And when you go to the page and you see, okay, this day we talk about MHC, and there will be the learning objectives, what you need to know at the end of the day when you study this. There will be given chapters from the books. And me and Dr. Boone, we've decided that we give 2 books actually as the basics, not only 1. There will be more if you want to read, but these 2, what you can definitely rely on. And there will be given even certain chapters which you can read. You will have slides prior to the lecture. You can read chapters before the lecture, so you kind of already understand what we're going to talk about. You can easily, during the lecture, catch the flow. There will be recording, so you can go back. Also, we can talk about that during the lecture. If there are questions, feel free to ask any question if something is wrong. The instructors' emails are given at the top of the description of each module, so you are very welcome to message and ask questions saying, hey, well, you know, I really didn't get that idea. Can you please explain me again? We will be more than happy, because at the end of the day, what we need, we want everybody understand the concept and use it later in your practice. And I will be absolutely happy to answer any question in email. We even can maybe organize a very short meeting so I can explain and everything. So all tools are given, and given not after, but prior to the lecture. So you can always be on track and catch the flow.
So I know for those of you who are applicants, you might want to take that information well in hand when you're thinking about creating your study plan and the number of hours that you will need, just based on the amount of information that Dr. Lykina is giving you. And for those of you who will be beginning the program in January with the Introduction to Medicine course, keep this level of content and this level and amount of studying in mind as you go through your Introduction to Medicine. So we are out of time for today, but I really want to thank Dr. Lykina for spending this time with us. She and I are on very opposite sides of the globe, so it's wonderful to be connecting in this way, as well as with all of you from various areas. And thank you so much for sharing that with us at the beginning of the session. So if you do have further questions for us, you can get in touch directly with me, Dr. Nicolette McGuire, Director of Student Affairs. You could even email Dr. Lykina if you have questions about her lecture or about the Immunology E-Foundation. And as always, please do get in touch with your admission counselor or your admissions team lead for further information and if you have questions related to admissions. So thank you so much for your time and attention today, and I look forward and hope to see many of you or all of you in our classrooms soon. Bye for now.
Thank you. Thank you very much. See you in January. Happy holidays.
Happy holidays, everyone.